So what I would like to share a little bit is some of the work we're doing in terms of trying to model eating. I thought we're talking about foods. The first thing is a nice area to model is what happens when you eat. Uh, right. So I'm Serafim Bakalis. I work at the University of Birmingham, and oops, you can see Birmingham is roughly in the center of England, of the UK. It's called the Midlands. Tolkien actually was from here, hence Middle Earth comes from Midlands. I'm based on the School of Chemical Engineering, and it's been a very traditional chemical engineering department. You can see here some pictures from the early 1900s where people used to build oil rigs, and actually we started from oil engineering and refining. Then chemical engineering came along, and about 2010, we realized that the majority of our students didn't work anymore for the petrochemical industry. They actually worked for a lot for the foods industry, some for pharma, but more what we call structured products. So we started developing the area slowly. I joined the university in 2005, actually. And since then, we have about 5 academics working in the area of food engineering with 50 PhD students. So it's quite reasonably big. And there are 2 centers, one of innovative manufacturing and the other one in sustainable energy uses. And what I'm presenting is slightly different from manufacturing, but it's exactly the same principles. So why study food digestion? What is my interest in understanding what happens when food eats? And one of it is, as an engineer, probably the first reason is I was taught to how to design processes with very clear design criteria. When it comes to food, it's really not very obvious how you design foods. What are your success criteria? What are you trying to achieve? As a result, so this is one personal, if you want, aspect of it, of designing foods, but the more specifically is designing healthy foods, and obesity becomes a major academic. When we say at Europe, more than 20, on an average, I would say 20% of the people are obese, and that brings significant challenges in terms of health. It's food-related diseases. It's becoming a major problem from Europe. And when we looked into what is the cause of obesity, what causes some of the foods to induce obesity, I realized as an engineer slash physical scientist that the mechanisms that they're taking places are not very well understood. So I started looking at the humans as a digestive machine. So if you see a typical graph, you have different parts. You have an oral processing, you have a stomach, and then you have a small intestine and large intestine. And you can make a graph actually that looks as you breaking particles down and then goes to a whole of tubes that the reactions take place. So that, and I used to take petrochemical engineering, actually looks like a chemical plant. So the idea is to use chemical engineering expertise to map the human as and use process engineering approach. So what do we know about the process? And this is my daughter trying to eat. It's something that we do inherently from a very small age without really much thinking about it. Uh, Typically, as most of the things, everything started in Greece, and it is kind of a joke, but actually, when we looked at who were the first to think about digestion, you can see Hippocrates and Aristotle, and they start mentioning things that are very, very clear, is still relevant today. For example, about lubrication, about mastication. Then we go in the Middle Ages, Leonardo da Vinci, and in iatrochemists first coming during the Middle Ages. So this is the first time where chemistry starts becoming important, and we talk about fermentation. And then when we go to more recent times, in the 1600s, there were first people who would try to taste actually things and do experiments, and things that today we wouldn't be able to do as a result of health and safety. So one of the first studies, quite consistent, was done in the 1800s, where Adolf Buchmal found somebody who could do sword intubation. He was a sword swallower, so he was able to swallow swords and take samples. And much later, William Beaumont found a person, Alex Martin, which had a hole, and actually the contract wrote that Alexis would have to obey, suffer, and comply with all reasonable experiments. So I think what I would like to get you from here is a lot of people have tried to do digestion, and there's a very long history. One of the problems is it's not very easy to study. The process equipment, unlike most of the process equipments, are kind of difficult to get access to. So what you usually do, and that's an experiment from the 1940s, which is still relevant, you need to insert a 7-centimeter tube into the gut to take samples. And what you do now is something that, the results are still very relevant. You're looking at digestion of proteins. One of the things you tend to see is that there is a very big variability, especially from here. And that induces that whatever we're trying to model, there should be a big variability between humans. And this is something we need to account. So when we're designing foods and when we're trying to predict the performance of foods, that's something inherently we need to understand. The technology has moved on and there's non-invasive techniques. So that to get an idea, that's a video fluorography. So that's an X-ray where you can see somebody eating actually a cookie. So you can see the cookie here, and this is what happens in the mouth. You're breaking it up, you're moving your tongue, you make it into a bolus, and then slowly you would see here's the bolus swallowing. So this is quite a complex process with open boundaries of fluids with very, very non-well characterized properties. And here what happens in the stomach. So this is your stomach, it comes up, this is the last part, the antrum, and you can see the whole geometry is moving and it's opening and closing, moving fluids a little bit to the left and to the right, and you can see some peristalsis now happening. So in the small intestine, there is three parts. There's the first part, the duodenum, where the majority of the absorption takes place, and then there's jejunum. So you can see all these irregular motions, and there's two aspects of it. One is actually pushing the food, and the second is mixing the food. And you can get very well mixing experiments, which is what you get here. If you could see, actually, these experiments have done after 72 hours, which means, there was food in somebody's body for 72 hours. And there's a lot of work now recently done with MRI, where you can start getting phenomena occurring, for example, in the gastric environment, in the stomach, that they would be very relevant to phase separation. Now, this gives you an idea of the complexity of the motion and some idea of what is actually taking place. If we start looking a little bit at the length scales relevant to what I call the digestive machine, in terms of the small intestine, your small intestine is a very complex biological structure. It has a diameter of a few centimeters. When you go very close to the wall, you see these villi structures, which are done to increase the surface area. And if you go to each one individually, you start going to sub-micron scales. So really, we're talking phenomena that they span quite a few length scales. Now, the villi can actually affect the flow because the way they move by contracting, they induce a secondary flow that enhances mixing. So the coupling, formally, should you want to address it, it should be a two-way coupling. Why we want to study food? This is one of the reasons why. This is the basis of most diet drinks. So the idea is that if you increase the viscosity, and this is a drink with guar gum, with increased viscosity, you drop the postprandial glucose, which is the glucose you have in your body. So actually you can control, and there's strong evidence of that, of GI, you can control your GI, your glycemic index, by changing the food structure. So as food process engineers, this is what we would like to do. We would like to produce foods that are healthier, that they have a smaller GI, that therefore would decrease the chance for type 2 diabetes. So there is the potential. What we need to understand is how much of that you should increase the viscosity and what are the mechanisms that will drive it. And when we try to understand the mechanisms, they're still not very clear. People talk about enzymes, going into a matrix, breaking the matrix down and making the nutrients available. So there's something about how the physical structure, the integrity of the structure interacts with your digestive juices, your enzymes, your bile salts, your emulsifiers to release the nutrients to make them available. So what we've been working is not as much with the biology, but as much as what are these mechanisms, what are these reactions, and what is the rate that you can make things available. If we go, one of the things we try to do is try to rationalize a little bit the phenomena. So in essence, your characteristic size of your foods goes down as you go down your column. So you have different fluids ingested at different compartments of your body. So in terms of oral processing, there's a whole range of phenomena taking place. And usually the characteristic length scales for oral goes from centimeters to micrometers, which is something that happens close to the tongue. In terms of your gastric environment, your stomach, we're talking about a liter roughly. So you have mixing with fluids, reduce the size, change the pH. You have a lot of enzymatic actions and a little bit of absorptions of minor components. Intestine becomes much bigger. We're talking about 5 liters capacity, and it can be about 7 meters long. So there you have again mixing. You increase the pH. You have a lot of enzymatic actions that can happen either in the interface or in the bulk. You have a diffusion phenomena through a boundary layer, a transport through a mucus layer, and finally an absorption. And what is fascinating here, you have all these interacting mechanisms, and it's not clear if there's one controlling mechanism. And these are some of the questions would be interesting to answer. In the colon, you have very gentle mixing and mainly fermentation, and then finally control absorb water. So your body tends to control water. You tend to put water on the gastric phase and reabsorb it on the colon. If we look a little bit onto other industries that are interested in this area, pharmaceutical is one of them, and there's a very strong industry. There's actually regulatory requirements for pharmaceutical because you want to control the dose that you deliver, and especially for drugs that are insoluble, you might not delivering what you say on the packets, or some cases you might over deliver. So there's USDA approved apparatuses that people are used. And actually, the difference becoming very, very small in some cases. So it is quite fascinating how these have been designed and been validated. In terms of modeling, the main mechanism that people are interested in pharmaceuticals, it's the crystallinity and some dissolution profiles. So people have come up with some idealized phenomenological models. And based on that, there are packages that you can do equation-based of the dissolution and the transport properties, and these are commercially available and commonly used from the pharmaceutical. And then when you go to the next stage, you can have compartmentalized models, so you have something absorbed in your stomach, and then you can find out how it's been distributed throughout the body. And eventually, what you really want is you dosing, you have doses of an active ingredient, and then you can find out what would be the effect. What would be the circulation of the drug at the required location. So now we're trying to get a little bit more into the modeling approaches, and hopefully I gave you a little bit of the background by now. So some of the major challenges is that the mechanism of the phenomena are not very clear, and quite often it's not very clear what you're trying to understand. And the key problem is to really link among different length scales. We'll start for the mouth first, and we'll present some key works. So some of the early work is done on the mouth, and that has to do really with the sensory experience, is how can you, by consuming different amounts of liquids and soft solids, what is the right rheological properties? And here we can see that there could be a range of rheological properties, but sometimes it's the sear, sometimes it's the stress, depending on which part you are. In terms of the mechanisms, the science has moved on a little bit. So there's different mechanisms that happen. So you can fracture. When you go into sensory perception, there is a lot of work that needs to be done and a lot of understanding on how your tongue perceives the fluids. So these are very narrow gaps where tribology takes place. And then we can go into combination of rheology and tribology. And going into tribology, there's evidence earlier from the 70s just to understand, and this is one of the modeling approaches. You can simplify the geometry of having a tongue touching your mouth, and then what you're interested in, what is the rheological properties here, and how do they correlate with some sensory experience. And by correlating, for example, with smoothness, with the friction force, you get an effect on viscosity, or thickness, for example, with the shear force, you can get a different relation with viscosity. So the modeling approach here, parts of the modeling approach is really coming up with scaling rules. Some of the work I've done in this area is really modeling oral processes. So using, again, a process engineering approach, where you have the mouth almost as a stair tongue, you have the different layers, and your sensory experience depends on how quickly the salt reaches the taste receptors. So the dots with the standard deviations are the experiments. And what we've done is we built different mass transfer equations, and we used some of the mass transfer relationships that would be known from from the process industries, and we're able to do a priori reasonably well predictions, as well as reverse engineer how would be the effect of different structures when different viscosities. So this is something that would allow us to start thinking about how can we influence, what do we need to influence sensory experience. And if you're aware of some of these ideas actually are not very different to some of the technologies coming out in terms of sweetness and sweetness enhancers. Moving a little bit further on, some of the other modeling approaches that can be done is Benjamin Lereveran, Nestlé, and NRC has taken over the approaches and continued some of the work. Try to understand really, going from a first principle, liquid structure interactions on what is the mechanical properties of the papilla you have on your tongue, and how this allow you to perceive shear and normal stresses. And it's been showing that the form of the papillae, they really can enhance the experience. And Maria Kalabidis in Imperial, who actually is part of the cost action, is doing a lot of work on the mouth and trying to understand what are the fracture mechanisms and how this related to the phase down, to the properties of your foods. Moving a little bit further into the small intestine, into the absorption, what I was interested in is not really new science in many ways. It's been reported back from the 1890s that mixing and motion is important. So this is a realm where chemical engineers have something to contribute. So the idea here is that if you have kinetic limitations, for example, mixing and absorption, then you can delay the rate of absorption. So this is a not fully explored hypothesis. So what we've done experimentally, we built it a rig that you can control the motility on very accurate way, so you can measure mass transfer. And what we're seeing by measuring an overall mass transfer coefficient, that there's a certain concentration of thickener you're putting on the bulk of the fluid, and after a while, the mixing is not very good. You start not having any true mass transfer. And why this is happening is because you start getting so viscous materials that you're just not having anything reaching your membrane, reaching the size to become available. Taking a step further, we look at different fluids, different concentrations, different viscosities. So what we're finding it out, and as you increase the Reynolds number, there is an area where you start moving slightly away from your laminar regime, and that gives an incredibly increase a lot the mass transfer coefficient. So we also took the step further then on trying to use some computational fluid dynamics for a two-phase fluid, for one-phase fluid, coupled with some reactions. The challenge here is that this is a fluid structure interaction, so you apply a pressure, you move the fluid, and then the normal pressure has to talk to the pressure that you apply outside. So one of the interesting things we're finding, as you increase the viscosity, and at viscosity 0.01 Pascal seconds, there's an area here where we can see a recirculation. And this is the recirculation that we believe enhances mixing and makes things more digestible, gives us essentially higher GI. Going to other approaches into modeling the gastric and the intestinal environment, there is work done on the stomach and similar CFD work done on the small intestine. These are some classic papers. It was very interesting recently, people trying to couple the micro scale mixing. So this is the papillae I mentioned that they've been moving and therefore inducing some flow. So that was a lattice-Boltzmann simulation. And then this can be linked to what is the effect of the bulk scale. So there's a whole range of fluid dynamics that they can be done, although some of the assumptions, well, you need to certainly make assumptions to oversimplify, but it all comes to conclusion that there's a flow and a mixing effect. When we go actually how we eat, this is a quite complex mechanisms where you have sensors. So something is coming out from your stomach, there's sensors that induce higher motility, or you stop eating, or you stop emptying your stomach. So what I'm trying to expose you here is that the gut doesn't operate in isolation. It actually is a system that operates by itself, and it's very, very closely controlled, and there are a couple of control mechanisms. So what we need to do as engineers to try to understand that is try to come up with a simplification of the system. So this is what we're mainly interested on, what happens on the gastrointestinal tract, and that can feed up to a complex range of phenomena. So the first thing we're interested is really liquid meals, something of different viscosity like a yogurt meal, and the idea is you're eating something, so they shouldn't have a structure that you need to break down. So the process model we're using is we're building process models where you put something in your stomach at time zero, about 75 grams of glucose. Some of it has been transported to the intestine, and then it's been absorbed. So the stomach we model as a first order, the stomach emptying, the gastric emptying, as a first order reaction kinetic with some half-time characteristic, and this is something the medics are very well aware of. The small intestine is a convection diffusion equation, and there's a change of mass. This is the advective term, how much you're moving along. And finally, there's a reactive term, which is what you absorb. In terms of the reaction, you have three mass transfer coefficients, something in the boundary layer, something in the membrane, something in the blood. And in essence, we assume that the main control is whatever makes available. So really what we're interested in is what we call bioavailability or bioaccessibility. The mass transfer coefficients, we take them from standard relationships of Sherwood versus Reynolds, and that allows us to calculate mass transfer coefficient a priori as a function of diffusivity, so essentially viscosity. So we're doing the work, and from that we're allowed to find out independently the effect of gastric emptying time with the effect of the viscosity. We identified that there are four regions onto what controls the rate of absorptions, and interestingly, most liquid foods would be here. So most liquid foods, the absorption is mainly limited by the luminal mass transfer, the mass transfer in the small intestine, which gives us a leverage that if we can change the viscosity of that liquid meal, we would change the mass transfer, and therefore we would delay some of the absorption. We then moved a step further, where, as I told you, the gastric emptying is controlled with some rates of nutrient sensing. So we put a feedback control mechanism, and then we start finding out when is the feedback control kicked up. And there's quite a few theories that the nutrition the medics have. So now we start able to explore a hypothesis. For example, you need to have the feedback mechanism is only kicked in once you start having a lot of glucose. And then in terms of viscosity, that can have an effect on to how quickly do you sense the nutrients. The interesting thing comes when you start integrating the whole system. Now, this is very important because there's a lot of hypotheses going on to what is the effect of sugar, and especially the sugar that is very quickly released in terms of inducing type 2 diabetes. So how much pressure are you putting into your insulin systems? There's quite a few models that most of them are equation-based. Actually, all of them are equation-based. And interestingly, this is some of the work also we're doing with Jan van Imbe from Leuven. And interestingly, a lot of the systems of equation, the stability of them is not clearly very well understood. So when we start trying to model a dynamic systems, the number of equations was quite high overall. So we start having problems with stability, and this is something we really need to look a little bit further on. For very simple fluids, we managed to develop some correlations with no delay and delays on the response time of the systems. So our models start comparing reasonably well with the literature. So we're confident that it's something we can build on. So start trying to close where we're going to plan to take this work. We've done some process modeling. We start looking at the dynamics of the systems. We've done some CFD to understand the kinetics. One of the things we realized that this is really an interdisciplinary multi-scale problem. So right now we're starting a UK-based consortium that is funded from BBSRC, where it has scientists from different universities. So we'll take you over what some of Birmingham's contribution would be: process modeling, which is a centimeter scale, and CFD approaches. On Imperial, there would be two people, Maria Karalabidis, who's going to be doing a lot of fracture mechanics. You can see some videos on the finite elements he's going to do. And that would help us understand some of the breakdown, physical breakdown of the matrices, that then they're gonna lead to what is happening of the mucus layer. So Julia Yermans from Oxford is doing some mesoscale measurements on how polymers and long chain diffuse into gel networks. So the idea is we're going to be able to go within one program, try to start integrating, and this is a proof of principle, centimeter to micrometer scale. This is going to be one-way coupling. The program actually, interestingly, is run from Imperial. And one of the key points is that Gary Frost is going to be doing some biology to allow us to validate some of the experiments with actual real data. IFR is going to contribute as well with us to do some in-vitro experiments to validate the models. And also University of Leeds with Alan Mackey, who's going to be mostly interested in absorption to mucus layer. And then we also have a few industrial partners that would help us design some model food matrices that we would be able to test. So in principle, that links some of the modeling techniques you would need to integrate in order to start understanding how foods perform. I usually see it as the opposite process of structuring, which it's not thermodynamically the same process, not the same drivers, but you're following the same path on the phase diagram. So eventually what we'd like to do is understand the sugars that develop in the receptors. So overall, in terms of the future, complexity, I think it's something we need to understand and we need to integrate. And complexity goes further beyond to people who do medical, who understand really the system, who understand the biology. And this is something we need to integrate. And these are some examples. So, and we need to work across different length scales because the controlling mechanisms are not very clear at all. So in conclusion, there's different model approaches you can take to understand parts of what happens when you digest, when you eat foods, and it all matters of what you want the answer to. It's very good to have a very clear problem definition, and as typical, as a good modeler, you should have a very clear statement of the assumptions. You should understand what are the interactions between different scales, and understanding, even finding what is the controlling mechanism is quite clear. And overall here, sometimes people talk about also about the in-vitro and silica model. And then, but in essence, the modeling development is quite generic. It has to do about what you're interested in. What is the question you're trying to answer? If it is about food structure, really the model should be evolving around food structure and what is the effect of. And this is an artist interpretation of a digestive system. So with that remark, I would like to thank you.